Cardiovascular diseases represent the major cause of morbidity and mortality worldwide and are the major cause of death worldwide, claiming about 18 million deaths annually. Meanwhile, current clinical strategies, including drug therapy, surgical interventions, and organ transplantation, offer limited benefits to patients without regenerating the damaged myocardium. Multiple studies have been conducted so far in order to develop treatments able to prevent the progression in patients and to treat the clinical courses in pre-illness persons at risk of those pathologies. Despite the progress, current therapies are still hampered by poor translation. The valuable results into clinical cardiology related practice. Cardiovascular disease, in particular, myocardial infarctions, MI, has become the predominant cause of human disability and mortality in the clinical settings. Several approaches were tested to regenerate infarcted heart muscles with minimal success due to the limited improvement of necrotic cardiac tissue following the re-enhancement of blood flow in the infarcted parts of the myocardium. This proved to be attributed to the restricted regenerative capability of the heart muscle and the rapid formation of fibrotic non-contractile scars. Well, among the present-day alternative approach, it's now focused on administering exosomes being rich in miRNA and mRNA to enhance the angiogenesis process and to favor their therapeutic application for the regeneration of cardiac muscle following myocardial infarction. Look, it's just an illustration in brief, exosomes being enriched in miRNA and mRNA, which are able to secure cardiac repair. This is kind of the cycle interpreted just below. The application of exosomes have paved the way for the alleviation of cardiac tissue injury along with conventional medications in clinical practice. No comments, but heart injury such as myocardial infarction leads to cardiac myocytes loss, fibrotic tissue deposition, and scar formation. Those changes reduce cardiac contractility, resulting in heart failure, which causes a huge public health burden. In the past decades, studies have focused on mechanisms underlying the regenerative capability of the heart and applicable approaches to reverse cardiac injury. And preclinical and clinical interventions show the potential to reduce scar formation and enhance cardiomyocyte proliferation. that counteracts with the pathogenesis of heart disease. Moreover, recent unique advances in developmental and stem cell biology, stemness, have switched on a grand green light for a wide spectrum of principally novel cardiovascular therapies, from the transplantation of stem cell derived cells to the stimulation of endogenous regeneration process. A special accent has been put on stem cell driven therapy, which has emerged as a promising approach to promote cardiac repair and improve cardiac function. That approach has opened up new perspectives for treating myocardial infarction and even for preventing the latter. But cells transplanted from the ionic heart often show poor recruitment and thus survival. Just look, in this context, regenerative medicine, I will stress, regenerative medicine globally, is just the field concerned with the repair and restoration of the integrity of damaged human tissues as well as full organs, including cardiac tissue. Stem cells have received significant attention in preclinical studies and clinical trials. And apart from the known potential for differentiation into the various body cells, stem cells enhance the organ's intrinsic regenerative capacity by altering its environment, whether by exogenous injections or introducing their products that modulate or modify endogenous stem cell function and fate for the future and sake of regeneration. The major drawback of such strategies is represented by the limited regenerative capacity of the cardiac tissue. Hence, the ability of the heart to recover after ischemic injury depends on several molecular and cellular pathways, and the imbalance between them results in adverse remodeling, culminating in heart failure. Look, please, for instance, mesenchymal, MSC stem cells were proven to be effective in direct and indirect differentiation for myocardial regeneration, angiogenesis, and cardiac functional restoration globally. The use of MSCs in regeneration of the cardiac myocyte and vascular endothelial cells of cardiac tissue is encouraging. MSCs have been established as one of the potential therapeutic agents following several tests in animal models and even in clinical trials. MSCs can protect the myocardium, the human myocardium, by reducing the level of chronic inflammation, promoting the differentiation of myocardial cells around infarct areas, and angiogenesis, increasing apoptosis resistance, inhibiting fibrosis, which are ideal qualities for cardiovascular repair. Meanwhile, through MSC-driven therapy has achieved considerable admiration. Some challenges still remain that need to be overcome, to be surmounted in order to establish it as a successful and productive technique. Look, challenges. Use of MSC for cardiac regeneration. In the context of MSCs in cardiac regeneration, some pathways and processes might exist that still remain unknown, still being unexplored. The other challenge is the prolonged survival of MSCs. Post-engraftment into the host myocardium. Pluripotent stem cells, including embryonic stem cells, ESCs, and induced pluripotent stem cells, iPSCs, have the potential to generate all tissues of the human body, including cardiac myocytes, offering a unique potential for cardiac repair and the PPM-guided regenerative medicine. ESCs and iPSCs represent an ideal cell source with the potential of unlimited expansion, differentiation into desired cell types in a controlled manner, and well-established functional benefits upon transplantation. In the complex and much complicated scenario, some promising chapters of regenerative medicine have been opened and being still opened within the discovery of pluripotent stem cell, PSCs, generated from patient-specific somatic cells and providing an autologous source of human cells. It's just the illustration of the above mentioned. Particularly, human PSC cardiac myocardium cells have shown robust potential for regenerating injured heart. And the protocol to differentiate HPSCs into mature cardiac myocytes at high efficiency have been developed, opening the door for targeted clinical application. It's just the illustration of immune-staining images of human pluripotent stem cell-derived cardiac myocytes obtained by confocal microscopy. Just the proof. iPSCs as the novel and moreover individualized pluripotent stem cells have been introduced as the appropriate candidates for personalized cell therapies because of person-to-person and population differences in the result of stem cell therapy. Individualized cellular therapy must be adjusted according to the patient's specific profile, including genomic and related ones, in order to achieve best therapeutic results and outcomes. At present, HPSC cardiac myocytes are the only realistic option of meaningful remusculization of injured heart. As you might see from the above mentioned, a population of pluripotent stem cells, including embryonic and iPSCs, have emerged as an attractive candidate stem cell source for obtaining cardiac myocytes because of their tremendous capacity for expansion and unquestioned potential to differentiate into mature cardiac myocytes. PSCs were proven to be able to efficiently differentiate into mature population of cardiac myocytes. Briefly, PSCs can be generated from the inner cell mass of the blastocyte, from a normal fertilized egg, from an egg that has undergone somatic cell nuclear transfer, or by engineering, reprogramming somatic cells with pluripotency factors. So, PSCs represent an ideal cell source with the potential of unlimited expansion, and thus unmanipulated PSCs, or those that have been genetically modified, can be used for drug discovery and drug testing, cell replacement therapy, or modeling human diseases in culture. Cell therapy using PSC cardiac myocytes tandem represents a durable therapy for heart failure in the very near future to come. More precisely, for heart repairing applications, a greatly scalable and medical grade cardiomyocyte generation must be developed and used. However, results obtained from clinical studies are inconsistent, and stem cell based improvement of heart performance and cardiac remodeling was found to be quite limited, and is not available to be broadly practiced. The key problem is to identity of cells be born to generate functionally active cardiac myocytes, replenishing those being lost during post-infarction ischemia. In order to optimize stem cell efficiency, it's crucial to elucidate the underlying mechanism mediating the beneficial effect of stem cell transplantation. Based on those mechanisms, biodesigners and bioengineers have developed different improvement strategies to boost the potency of stem cells repair and to generate the next generation of stem cell driven therapeutics. Just look, it's biodesign engineering strategies to accelerate clinical translation of SC stem cell driven therapies into the genuine practice. As you might see from the above mentioned illustration, the combination of the paracrine effect and the potential activation of the intrinsic program of cardiac cell, which is triggered by cell-cell crosstalk, are our top priorities of the next decade of advancing stem cell therapy for heart repair. So new cell-based therapies have been intensively studied in the last few years due to the potential benefit in patients with acute myocardial injury, stable coronary artery disease, and heart failure. The phenomenal achievement in this area is the identification of resident cardiac stem cell, supposed to be a crucial source to initiate and prompt myocardial self-renewal and regeneration, but can be developed inside the mature cardiac cell by formation, as I already told, cell-in-cell structures, CICs, which in turn being encapsulated are involved into cardiac myogenesis, thus opening up a green light to secure the targeted management of post-infraction regeneration. So, cell therapy using PSC-CM tandem represents a dual therapy for heart failure in the future. I should tell you that a combination of canonical stem cell regeneration, omics-related technologies, nanobiostructures, and the armamentarium of special IT algorithms would be necessary to be consolidated and integrated to develop a principally new platforms to utilize the resources and move ahead very, very high speed. So the market of the application of stem cell therapy in the cardiac practice is still very young. And a transdisciplinary team would have to develop a special initiative to move ahead together as the solid team of the next step generation. Thank you so much.